Professor Rawlings, ladies and gentlemen. I'm very pleased to see you all here, and I'm delighted to be honoured in this way by the university to be invited to give the Oxford-London lecture. As you heard, I'm going to be talking about a pretty topical subject, and one which, to be absolutely frank with you, I knew very, very little about 2 years ago. And that is part of the point. Ebola took us all by surprise, not because it hadn't appeared before, but because of the way that it did appear in West Africa in late 2013, early 2014, and of course the global impact that that very serious outbreak had. So what I'm going to be talking to you about is a little bit about vaccinology, which is what I do as my day job, about what vaccines do, how they can help, and much more importantly, why we did not have an Ebola vaccine ready and available to use when this outbreak occurred. And the reason for that is not scientific, as I'll outline. It was because essentially we couldn't make a business case in the vaccine companies that make most of our vaccines and provide them to the world to invest in making an Ebola vaccine, developing it, spending probably hundreds of millions of pounds to get it licensed and available for use to protect people in Africa and elsewhere against this dreadful disease. So just a reminder, as you've heard, this is an unusually fatal infection. Around about 70% of people who become ill with Ebola will not survive. We have no licensed drugs. We have still no licensed vaccines against Ebola. And Ebola was used to popping up in Central Africa, in Sudan, but it had never appeared in West Africa before. So it was really unexpected in that region. The Guinea, Sierra Leone, and Liberia outbreak were unprecedented, not just in their location in West Africa, but in the scale of the emergency. We ended up with over 27,000 cases, over 11,000 deaths, and a global response that was truly impressive, but in many senses too late. We did have vaccines sitting in large bags available to be tested at the time of the outbreak. But of course that isn't good enough. You can't just go and open the bag, distribute the vaccine, vial it, and start immunizing tens of thousands of people with something that is literally untested in people. And that was the situation. None of the vaccines that went to West Africa during the outbreak had been in human beings before this emergency. I'm going to be talking to you today about a couple of vaccine types, principally about one which comes from chimpanzees, or at least the original vector of the vaccine comes from chimpanzees, and it's an adenovirus, the sort of virus that causes common colds in many of us fairly regularly. And then a second virus, vesicular stomatitis virus, one that's hardly been heard of by most infectious disease specialists. It's a vaccine that infects cattle occasionally, but is also useful as a carrier of antigens for vaccine design. And that bat skulking in the corner there is just to remind you that we think that the ultimate reservoir of the Ebola virus is in bats. It may be transmitted through non-human primates, through bushmeat that people consume in particular countries, but it's going to be difficult to eradicate Ebola because we're probably never going to go and vaccinate millions of bats to try and get rid of the reservoir. So this is just a map from Chris Dye at WHO of what was happening in the earlier part of 2014. You can see that there was a cluster of cases exactly 2 years ago in March. And most of the wise heads who had seen Ebola outbreaks before were pretty confident that, like all the previous outbreaks, this one was going to go away, or at least be controlled by isolation and the traditional public health measures that had controlled outbreaks in the Congo and in Sudan and elsewhere. And by week 17 or 18 or 19, it looked as if that might just work. But by May, it wasn't looking good because the Guinea cases had led to cases in Sierra Leone and then in Liberia. And then by September, people were seriously worried. By late October, people were literally panicking because the epidemic curve was still rising, and it didn't turn around until almost the end of the year. So what do? There was a call for research on vaccines. There was a call for testing new interventions that had not been validated or certainly not registered or licensed. We started using the small amount of monoclonal antibody that was available to treat small numbers of cases and trying to get into gear a research activity to test new products that was frankly very, very difficult because in these countries the primary goal, of course, was to control the outbreak and to treat patients. So vaccines, of course, I'm an enthusiast for. They are important. They save 2.5 million lives a year. They are a commercial success. This is a growing market globally, over $25 billion in sales every year. Vaccines have been and will continue to be key to disease eradication. Smallpox is gone. Polio is nearly gone. Measles is next on the list. A livestock virus, rinderpest, has been eradicated. Even measles, even malaria, there are ambitions to eradicate with the help of vaccines. And other things that are equally important, food security, maintaining our repertoire of antimicrobial drugs, biosafety, and economic growth are all to a degree contingent on the success of vaccines. So 10 years ago, in the University of Oxford, we decided to get serious about vaccine design and development. Prior to that, there had been lots of people working on vaccines, but it wasn't really coordinated in the way that we thought could make most impact. So we founded an institute called after the founder of vaccines, Edward Jenner, back in the 18th century, a country doctor in Gloucestershire, doing the first vaccination famously on his gardener's son against smallpox. So the Jenner Institute tries to bring together a lot of the talent in the university and the research groups that are interested in designing and developing new vaccines and leveraging the extraordinary strength in the biological and other sciences that we have at the university. And in principle, making vaccines shouldn't be all that difficult. We've made many. There are about 27 on the market. There are some that are proving extremely difficult, like HIV vaccines, for very particular reasons. But in general, what you need to do is to select what immune response you want, presumably against the virus or the pathogen you're trying to protect against, which bit of that pathogen you're going to put into the vaccine, and from a large pathogen like malaria, it might be just one antigen, or in some cases it's easier to select, as it was in Ebola. I'll come to that. And then you have to figure out how to deliver that antigen, which covers immunogen design. Do you want to use a peptide? Do you want to use a viral vector? Do you want to take a protein from the virus and make a strong immune response? And making a strong immune response is a large part of the game. So you often have to try different vaccination regimes, different dosages, different intervals, and that's all very important and proved important with Ebola. You do need to know a little bit of immunology to understand vaccinology, not a huge amount, and most of what you need to know I can capture on one slide, which is the good news. So most people who make vaccines try to trigger one arm of the immune response. There are two main ones, the humoral arm and the cellular arm. And if you're working on this side of the slide, you're trying to make antibodies, largely IgG, which are the most durable antibodies. And one of the great things about vaccines is they can last for years, even for your entire life, if you design a very good vaccine. And most of that protection is through antibodies. It's been much harder to trigger the other arm of the immune system that works through white blood cells, specifically lymphocytes, very exactly T lymphocytes, and they come in two flavors: killer T cells, which have a marker called CD8 on their surface, and are very good at tackling viruses that have got inside cells, and CD4 T cells, which have an important regulatory role. So you can look on this as antibodies mopping up pathogens outside of cells before they get in, And then if you lose here and your virus is already inside a cell, as it often is with, say, HIV, you would then need these T cells to come and kill the infected cell and the virus with it. So we have vaccines these days that can induce high titers of antibodies and others that are specialized at inducing cellular immunity. And one of the questions with Ebola was which of these did we need and which was going to be better. There are a variety of technologies. I won't go through many of them, but I will mention that to get antibodies, you often need to make a particulate vaccine. This is about 20 nanometers in size. And to get a cellular immune response, you need to get your antigen inside the cell, into the cytoplasm. And the best way of doing that is to use a virus, usually a crippled one, so it doesn't cause a disease, and you put your antigen into that viral vector. So this is genetic engineering. You take your gene, say, from Ebola, and snip out the DNA for the part of the Ebola virus you want to vaccinate against, and put that into the DNA of another virus, in this case an adenovirus, and you can then immunize with that and get an immune response against Ebola. And people were doing this in all sorts of interesting ways. One of the uses of an Ebola vaccine that was being pursued was to protect chimpanzees and gorillas. There were actually vaccine trials going on in a zoo before the outbreak. And this is because troops of dead chimpanzees are found occasionally in many African countries, and very often the cause of that is a small outbreak of Ebola in that troop. So conservationists had an interest, and still do have an interest, in vaccinating primates. But the vaccine I'll talk most about, that we did a lot of work on during this outbreak, was based on a very simple and now almost standard idea of taking a gene out of the relevant virus and putting it into a replication-defective adenovirus vector. So up here we have a cartoon of the Ebola virus. It's a very, very long, strangly virus, twice the length of a red blood cell, which is enormous for a virus. But usefully, its coat is mainly made up of one of its 7 gene products, the glycoprotein shown here. So most people were agreed on which gene or which part of the Ebola you would vaccinate against. And crucially, just putting in that one gene into a vector cannot cause Ebola. So this had been done previously. This gene had been taken out of the Ebola virus, put into a vector, as I will call it, a replication-defective adenovirus, and that had been used to immunize experimental animals. Another virus, which is again a vector, is this VSV, vesicular stomatitis virus, and a competing group in Canada. The adenovirus work was being done at NIH in North America. The Canadian group used this vector to put the same glycoprotein gene in, and this is a little used vector. It's not an obvious choice. It's of the rabies virus family. It had been shown to work in monkeys, which is important, but it is replication competent. In other words, it divides once you immunize somebody, so the virus grows after vaccination. And it had only been given to 2 lab workers who were unfortunate enough to prick their finger in a high containment lab working on Ebola virus. So whether this actually worked for them is not clear, but they survived. And this had been developed by a tiny company in the States, New Link Genetics, licensing it in from the Public Health Agency of Canada. WHO then inherited it, and finally Merck ended up developing it during the outbreak. And viral vector technology is something that we've worked a lot on at Oxford. It's often said that science, of course, is very good for emergencies. Well, in this case, the emergency was quite good for a particular part of the scientific work that we do, because suddenly these viral vectors became very mainstream and widely used for this particular outbreak. So 6 viral vectors were taken into the clinic, and almost all of them, 5 of them, went to Africa, and there was just 1 vaccine of another type that was developed in parallel. We know a lot about the target antigen. People had not only purified that glycoprotein antigen, they had solved its crystal structure, which is shown here. We knew which parts to target. There were monoclonal antibodies available in very small amounts that could be used to try and treat individuals. But the key data that led to our clinical trial in August of 2014 with ease. This was work done at the NIH by Nancy Sullivan's group, who had been working on Ebola vaccines for about a decade, funded, interestingly, by a biodefense program in the United States. So the funding you could get 5 years ago to make an Ebola vaccine was not to prevent outbreaks in Africa, unfortunately, but it was to defend North Americans against a potential bioterrorist attack. And there has been substantial funding for biodefense vaccine research, particularly in North America. So here is that adenovirus vaccine. It happens to be a strain from a chimpanzee, which is helpful because we don't have antibodies from our natural infections. And inside is the Zaire strain glycoprotein gene. Now remember, there are two main strains of Ebola virus. The one that was causing the outbreak in West Africa was the Zaire strain, and that's quite different from the other main strain, the Sudan strain. I'll come back to that. But critically, a single dose of this vaccine given to non-human primates, macaque monkeys, gave a B-cell response, that's the antibody arm of the immune system, and both types of T cells, good levels of CD4 T cells and very good levels of CD8 T cells. So that looked quite promising, but of course the key test is whether it works, and you're allowed to infect animals with Ebola virus, and when that was done, the efficacy with both of these adenovirus vectors was 100% in a small number of macaque monkeys, challenged or infected soon after vaccination. So that tells you it works for a few weeks. What about a bit later? Well, you wait, and then you infect other monkeys later on. And at about a year, the protection was waning. It was about 50% rather than 100%. But importantly, by giving a second dose of vaccine, actually a different vaccine called modified virus Ankara, which is a boosting vaccine, you could bring the protection back to around about 100%. So here we had data that these vaccines were not just safe in monkeys, but they were giving protection not just against Ebola infection, but actually a very, very large dose of Ebola virus. And I'll come back to that. Interestingly, you could try and dissect out the mechanism of protection. Was it antibodies? Was it T cells? Both are probably important for this vaccine. And you can see here that by depleting the relevant types of T cells, CD8 T cells, you lose most of the protection. But the antibodies are probably playing a part as well. And with the other vaccine, the VSV vaccine, efficacy appeared to be almost entirely antibody-mediated, probably because the VSV vaccine does not produce much of a T cell response. So we had evidence that there were vaccines that had been developed in animals that looked promising, that were giving high-level protection, both the VSV and the adenovirus, with a single dose. So how could you ignore that if there was an escalating outbreak in West Africa with thousands of deaths? So a lot of partners came together very rapidly at the beginning of August. I was phoned up by WHO asking how quickly we could do a clinical trial, and very helpfully by the director of the Wellcome Trust on the same morning offering to fund that clinical trial. And the team was as shown here. So we had 3 partners with GSK, who interestingly had inherited that chimpanzee adenovirus vaccine from a small Italian biotech company that we had worked a lot with, called Ocaris. GSK had bought Ocaris a year earlier, not for their Ebola vaccine. And of course, NIH, who had been doing those preclinical studies in monkeys. And our goal that was given to us was to start the trial as quickly as possible, to ask whether the vaccine was safe in people, as distinct from monkeys, and to try and trigger a trial in a more relevant population, if you like, in West Africa as soon as possible. You can't take a totally novel vaccine into a trial in Africa usually without doing trials in a northern setting. And of course, there wasn't much vaccine. There was one batch, a couple of thousand doses, but to do anything useful, you needed a lot more than that. So part of the grant from Wellcome covered manufacturing of this vaccine owned by GSK in Rome, trying to make 20,000 doses by the end of the year. So we were after safety. We were looking at what immune response we got in people. Could we get the immune response that seemed to protect macaques? And we knew what that was quantitatively. And then our deadline was the end of the year. We were already in August, September here, so by December, a decision had to be taken. given what was happening in West Africa, on whether to go ahead and begin to deploy this in a clinical trial to see if it actually worked, with a focus on healthcare workers who were considered at greatest risk. We managed to get started by the 17th of September and vaccinated 60 individuals very rapidly. We were overwhelmed by the response from volunteers who wanted to help. It was absolutely fantastic, and we were very pleased by that. And that allowed us to trigger the trial in Mali starting on the 4th of October. Now, for those of you who are not academics and don't do research all the time, let me explain that this sort of thing doesn't usually happen. If you write a grant application to any funding agency on the 14th of August, you are unlikely 12 days later to get a positive response saying, yes, indeed, we'll give you £1 million or more for your clinical trial. After 6 reviewers had reviewed it and 2 rounds of review. So that was a fantastic performance by the Wellcome Trust, turning that around in 12 days. We needed to get the vaccine filled. It was a new formulation targeting the Zika strain. We needed regulatory and ethical approval. The MHRA in London, the UK regulator, turned this around and approved it in 4 business days. The ethical committee met specially of a morning to consider our application. We didn't have to wait for the next meeting. So vaccines had to be shipped, to be labeled. The lawyers almost delayed us. They signed the night before on all the agreements, and we got started by the 17th of September, and were finished within a couple of months. But very importantly, WHO didn't just want data from the north, they wanted data from a relevant population. Where would you go? This was the exploding outbreak in the 3 relevant countries. As near as possible, but obviously not within a country. Very hard to do a phase 1 clinical trial if there are no healthcare services and essentially no research infrastructure. And we had a lot of options, mainly because we work on malaria. We have very good collaborators, for example, in Burkina Faso or in the Gambia, where there's a specialized infant immunology and vaccinology clinic run by the MRC. And the decision in the end was to go to the University of Mali at Bamako. This was our first volunteer, and the team there was led by Samba Sow, collaborating closely with Mike Levine at the University of Maryland. Now, of course, the key thing with these phase 1 trials is safety. That is your primary endpoint. You need to have healthy volunteers, you need to screen them carefully, you need to vaccinate them and follow them up very carefully, and this was our first volunteer. And we followed volunteers initially for 6 months. We're still following them 18 months later. And the safety profile of this vaccine has been very good. So in the first 92 Oxford vaccinees that we have looked at, safety has been very good, and so has it been in Mali, 91 volunteers, in Switzerland, in the USA, the usual minor side effects you would expect from a vaccine, with arm soreness, very rare fevers, and generally very well tolerated. So good, that was straightforward, and everybody was agreed that it looked safe. This was a little bit trickier. These are the immune responses measuring antibodies to the vaccine, with a low dose, a medium dose, and a full dose in group 3. This is the level before vaccination on day 0. There's the level 4 weeks later, day 28, which we took as the index time point. And the good news is that nearly everybody responds, 93% at a full dose. And these are the levels on the scale of antibody measurement, so nearly 500 on average units in the vaccinees, which seemed pretty good and many people thought was fine. Then there were the pessimists, and I was amongst them, who looked at the level of antibody in the protected animals, and to get sterile protection in macaques, you needed 3,700 units. So would 400, 500 do? Or did we need a higher level of immune response? And the short answer is nobody knew. There was the argument that the monkeys are given a much higher dose to challenge them or infect them than you might get from attending a burial in Guinea and rubbing shoulders with somebody who happened to be transmitting. But we didn't know that. What about the T cells? Were they any better? Well, the T cells were good, whether you measure them by ELISpot or by flow cytometry, 2 different ways of counting the number of T cells that are now specific for the Ebola antigen that you've put into the vaccine. But again, when you look quantitatively, the number in people was about 5-fold down compared to what it was in protected macaques. So the same problem really. It was good, but was it good enough? So one potential solution to that we actually followed up on is shown here. This is an immunization regime that we had developed at the university for a very tough target, which is a malaria vaccine, trying to induce very high numbers of T cells and antibodies. So in addition to giving a single dose of the chimpanzee adenovirus, you come back a month or two later and give the same antigen, depending on what you're vaccinating against, in the viral vector that is called modified vaccinia ankara, which is just a strain of the old smallpox vaccine. And we had done that for malaria, hepatitis C, HIV, flu, different groups at the university, and we expected that it should work for Ebola. Very luckily, people had already made that vaccine, again on biodefense funding. There was a lot sitting in Copenhagen at a biotech company called Bavarian Nordic, and they kindly agreed to provide it to us so we could test this hypothesis. And there was another lot that, again, Wellcome came in and funded a better matched insert to the Zaire strain. We made that on a cell line in fairly substantial amounts in record time. And very simply, of our 60 volunteers, we invited half of them to come back, 30 of them, and gave them a booster dose of the MVA, and the safety was, as we'd expected, rather satisfactory. But very impressively, this was good. We didn't think it would be that good. If you look at the antibody responses, and I'm now showing you a log scale on the y-axis, that's 100, 1,000, 10,000. There you are before vaccination, after the adenovirus, and then after the MVA, at 14 days later, your level has gone up 20-fold, way above the threshold that we knew was needed to protect those macaques. That's the antibody response. You can also measure the function of the antibody by asking, do they stop the virus infecting cells? We can do this in the test tube. It's called an in vitro assay, and it's called neutralization. So we can measure the neutralizing antibody titer. There it is after the adenovirus. There it was after the MVA boost. And again, it had gone up very substantially to levels that the reference lab in Marburg, who did this for many different trials, were really impressed by, saying they'd not seen the like of that before. So that was exciting. But of course, does it work in the real setting? And the group in Mali, again, performed extremely well and extremely quickly. Here they are testing on the top the adenovirus vaccine on its own. You get levels of nearly 1,000 with just a single dose. If you boost, it goes up to over 10,000, so a 36-fold increase. So in the relevant population in West Africa, this was looking very promising, not just for antibodies, but again, for T cells. These are the levels measured by flow cytometry of the critical CD8 T cell population, pretty modest after a single dose, much higher after a boost. So by this stage, we were feeling pretty pleased with ourselves. We had a vaccination regime that was safe, that had been tested in Africa, that was looking as if it was giving immune responses at least as good, well, really better than the level you needed in animals. The problem was that WHO, who were coordinating the whole activity, were less impressed. They said, you're giving 2 vaccinations, yeah, we don't really like that, but what's worse is you're giving them a month or 2 apart, and we've got an ongoing outbreak. Why don't you try them at, say, a week interval? And we did. We went back, redesigned the protocol, did the vaccination at a week and 2 weeks, compared it to the 3 to 10 weeks, which we had previously. And to our surprise and delight, the CD8 T cell responses were somewhat better, at least as good. And nobody knew that previously. We'd never done this for malaria. Antibodies, not quite as good, but certainly better than a single dose vaccination. So we felt that that was certainly suitable for moving ahead. And by now, we've vaccinated a lot of people, not just in Oxford, but also in Dakar, not just with the MVA from Bavarian Nordic, but a more matched strain to the outbreak strain of virus with very good immune responses and very good safety. Remarkably, at about the same time, we, meaning the university, and in particular my colleague Andrew Pollard and Matthew Snape, were approached by Johnson & Johnson, or Janssen, specifically here in Europe, who had been working for several years independently on making their Ebola vaccine. And they had come up with a very impressive protective regime in non-human primates, had excellent data, and said we wanted to take this into the clinic as quickly as possible, and we were pleased that they came to Oxford because of that. And what they came up with was not very different to what I've been telling you about. It's an adenovirus, Ad26, so a different strain of adenovirus, and the MVA, again, working with the same group in Bavarian Nordic. And they had data in animals showing that it worked both ways around. You could give the MVA first or the adenovirus, or vice versa. So that trial was started off as quickly as possible in December in Oxford. I think the first vaccinations were just before Christmas. And this has gone forward very rapidly as well, and trials have been done in Sierra Leone. And that strategy looks just as safe and just as immunogenic as the one with the chimpanzee adenovirus and MVA. So now let's just notice what's going on. We have now 3 of the world's biggest pharma companies. Merck taking on the VSV vaccine, GSK with their Ebola vaccine, ChAd3, and now Johnson & Johnson, all helping out, if you like, by developing vaccines, taking them into the clinic as quickly as possible, largely paying for that effort in the hope of being able to end this escalating outbreak in West Africa. VSV had done rather well. They had done a trial in Switzerland, in the USA, in Lambaréné in Gabon, in Kilifi in Kenya, Hamburg, Germany. They do see viremia in most of the vaccinees. It's a replication-competent virus. A lot become febrile, and there was an issue with arthritis, particularly in Geneva, but not so severe that the trials couldn't continue, and this vaccine went forward into West Africa very rapidly. And even though the safety profile doesn't look too great in Europeans and North Americans, it was pretty good when it got to Guinea. So now what happened? We're into early 2015. The good news is that in Liberia, things have turned the corner. The outbreak is clearly waning. Sierra Leone took longer, and Guinea never got up to the heights in terms of number of cases, in terms of case incidence that was seen in Liberia and Sierra Leone, but it dragged on much longer and was still going in the middle of March when this graph ends. So what was planned, and here's the grand plan, was to test all 3 of these vaccine types in large trials to see if they worked. That was itself not without controversy. Particularly early on in September and October, there was a very strong argument made that as soon as you had a vaccine ready, you shouldn't come and test it in these populations. You should just go and give it to everybody and use it in the hope that it would end the outbreak, which is an interesting ethical point. Of course, the regulators argued, on the contrary, that you don't know that that vaccine works, therefore you can't just go and give it to everybody. And if you don't do a trial, you won't know whether it works. You'll just see what happens. That isn't how you find out if your vaccine has any efficacy. And even worse, the pessimists pointed out that there have been vaccines that have made things worse. So the consensus that gradually emerged was that, yes, you needed to do controlled trials. If you do them in the right way, you monitor the data as you go along, the risk is relatively small in that the number of cases you would save or miss by not vaccinating can be controlled, and that's what ended up happening in Liberia. The design of a huge trial, 9,000 per group, 2 different vaccines, 9,000 controls, and you can see the vaccines selected there. Sierra Leone had another interesting design, focusing on healthcare workers in a so-called stepped wedge design, but neither of these happened. They started, but they had to finish because, happily, the number of cases fell so rapidly that the trials were not going to provide a result. But of course, the critical trial did happen, and that was in Guinea, designed by an international team, Norway, the United States, WHO, funded in part by the Wellcome Trust, and they decided to test the VSV vaccine first, and then the chimpanzee adenovirus second. And I'll come to the results of that in a moment. And Janssen had this ambitious plan to do a very large efficacy trial in Sierra Leone, but of course the case numbers fell there as well. So what did we end up with? A single trial for efficacy, but a fantastic result. Couldn't have been better in a sense. Now remember, ring vaccination is not something that you normally do when developing a vaccine against whatever, pertussis. You normally vaccinate half the individuals in the trial, you don't vaccinate the other half, and you follow them over time. But in an outbreak situation where you have a different option to control the disease by vaccinating around cases, ring vaccination, which had been very little used before, turned out to be very effective. So the idea is you identify a case as soon as you hear where that case is, you send your team out, you vaccinate the village or around that individual, depending on where they are, or as a control group, you delay vaccination. And in this case, the delay was a few weeks. The few weeks delay was critical because, as you can see here, in the 48 early rings, about 4,000 people, 42 late rings, there was a clear difference in the number of Ebola cases. If you vaccinated immediately, there were 0. If you delayed vaccination, there were 16. A nominal vaccine efficacy of 100%, pretty good confidence intervals, and a significant p-value. The vaccine worked from about day 7 to 10 onwards. There wasn't protection in the first week after vaccination, so no apparent efficacy in the first 10 days. But after that, for several weeks, there was this high level of efficacy. So the long-term durability is not clear from that, but we can learn more from that than the numbers would suggest. So this was unprecedented. A vaccine that had first gone into humans in September. The results of the phase 3 efficacy trial were published in The Lancet in July. I don't know of that happening before. I don't think it has for anything. So we can develop vaccines faster than we usually do. Are there lessons for developing other vaccines? A novel trial design. And immunologically, it was clear that a fairly modest level of antibody was protective in Guinea. So the optimists were right. You don't need 3,000 units of antibody. Probably 100 or 200 will do, which is great news, because that means that probably all 3 of the Ebola vaccines that I've talked about are highly effective, at least in the short term, comparing their immunogenicity data. So where are we now? The outbreak is effectively over. There may be the occasional recrudescent case. If you look at the numbers, there's a suggestion that the VSV vaccine used in the ring setting contributed to ending the outbreak. We can't say that with great confidence, but it looks very plausible given how effective it was. It's key to license that vaccine so it can be used in the future, and the other vaccines may be licensed as well. I'll come to that in a moment. So what we're doing at the moment is following those vaccinees to see how long the immune responses we induced persist, because we'd like to know how long the vaccine works for, and we're not going to have Ebola cases in Oxfordshire to be able to predict that, I hope. But the regime that looks most durable at the moment, to me, is adenovirus followed by MVA. You need 2 doses. And of course, we do need a vaccine for the other strain of Ebola, the Sudan strain. We only have the Zaire strain vaccine made and tested. Let's just notice in passing, coming near to the end now, that there are generally 3 phases of vaccine development. We do a lot of phase 1 and phase 2 at our institute in Oxford. And this is small numbers for safety in phase 1, then maybe hundreds in phase 2, and then a key phase 3 efficacy trial. What happened with Ebola was phase 2 was skipped. People went from phase 1 in West Africa, say in Mali, right into a phase 3 efficacy trial for licensure, and are now going back to accumulate the safety data that you need to license your vaccine in phase 2. So that was another quite remarkable part of the whole activity. And as I said, we are comparing immunogenicity and particularly durability of these immune responses. This is just at day 180. We've gone well past that now, and we can see which vaccines give durable immune responses, and it's all looking pretty good. But what about the vaccines that didn't get into a test of efficacy? How can you license those? Well, this is a more general issue, because for most outbreak pathogens, if we want a vaccine against SARS, hopefully we're not going to have a SARS outbreak anytime soon. How do you license a vaccine when the disease isn't around? There is a solution, an alternative route for licensure, both in Europe and in North America, where you need to demonstrate efficacy in a suitable animal model. Well, that was done early on for Ebola. Identify the immune correlate, or ideally mechanism, and show that when you vaccinate people, you get that immune response. And that's exactly, of course, what's been happening over the last 18 months for Ebola. And finally, you need an adequate safety database, not in hundreds of people, but in many thousands, and that work is continuing and in diverse populations. But the conclusion is, I think it's pretty likely that several of these Ebola vaccines are going to be licensed by the relevant regulators. And that's critical because, as I come on to now, we need to be prepared for the next outbreak, and part of that involves having a stockpile of suitable vaccines. So there are many lessons. One of the obvious ones is that we shouldn't have been doing this, not because we really shouldn't have done it, but we shouldn't have needed to respond in this sort of way in an emergency outbreak situation, trying to develop a vaccine in months that would typically take 5 or 10 years or more. We need some outbreak preparedness, not lots more rapid responses. And of course, we've seen issues before with SARS, with swine flu, with MERS, now with Zika, and so on. And I'm afraid the list is longer than that. This is a list of the outbreak pathogens that have troubled some countries somewhere in the last 10 or 15 years. You'll remember most of them. SARS, for example, Zika I'll talk about in a moment, pandemic flu, definitely, 2 near pandemics in the last 10 years. Chikungunya has spread widely, Rift Valley fever I'll mention. Many of you know about MERS in the Middle East, causing a respiratory syndrome, a virus not very different to the very scary SARS coronavirus that spreads by aerosol. And then other ones, enterovirus 71 in the Far East, Lassa in West Africa, that was endemic in some of the countries that the outbreak was happening in. West Nile, great panic about West Nile when it got to America from migratory birds. Vaccine development happened, good phase 1 trial published. All vaccine development stopped once the outbreak ended because there wasn't a business case for the company developing that vaccine to continue. So the problem that I'm here to talk about is that there are no licensed human vaccines for any of these. Well, you could stretch a point and say we have maybe a flu vaccine against some strains of pandemic flu, but the next pandemic, if it is pandemic, is going to be different. So why haven't we? Is it that we'd never heard of these viruses? Well, no. SARS was entirely new when it came along, but essentially all of the rest were in the literature, including Zika, of course, which hadn't seemed to be a problem but was known about since the 1950s. And of course, the simple answer to why there weren't vaccines is that nobody could produce a business case to persuade any vaccine company to invest the hundreds of millions needed to not just design and develop and make and manufacture a vaccine, but to do all those clinical trials and license the product. And that's the problem. We're at risk of some of these coming back, or new ones we don't know about. And we haven't got a mechanism whereby the main manufacturers of vaccines will get their money back. And many companies have lost money on the Ebola outbreak, stepping up, making vaccines very rapidly, putting some of their own resources and some of public resources into the program. And now it doesn't seem clear how that money is going to be recouped, if it is. So, need we worry? Will there be more outbreaks? Sadly, yes. Almost certainly. There are more people around, especially in Africa. Population growth is substantial. There are much bigger cities. That was one of the big problems with Ebola. The rate of transmission in Conakry and other capital cities was going to be much higher once it got there than it was in rural parts of the Congo, where previous outbreaks had been. There's obviously more long-distance travel, certainly by air. There are lots of viruses lurking. There are hundreds of zoonotic infections that we are aware of. Zika, we were aware of 2 years ago. We didn't know what it would do. And of course, new viruses will continue to emerge, just as SARS did. So my modest proposal about a year ago was that we could do something like what we'd just been doing for Ebola, which was to take a vaccine that worked in animals, and there are lots of these vaccines that do work in animals. Academics like myself do the studies in mice, show that the vaccines can work and look promising in a mouse, That doesn't tell you they'll work in humans, and we don't generally have the resources to even manufacture the vaccine for a clinical trial, let alone do the clinical development that's needed. But we know how much Ebola cost. Many of us were writing the grants, so we know how much that was. And to take a promising vaccine from animals into clinical development, do phase 1, phase 2 trials on public funding, and that's an if, using ideally a common manufacturing platform, you're talking about tens of millions, not hundreds of millions. And then what you would do is keep a vaccine stockpile where you could access it rapidly and use it to put out small outbreaks. And you wouldn't need hundreds of millions of doses, you'd probably need 10,000 for most outbreaks if you were quick off the mark, and moves are being made to have those teams ready to go. You would have emergency use approvals, you wouldn't have licensure, but you'd also get a read on efficacy in a ring vaccination setting. And this is not a novel idea. It's widely used in, for example, veterinary vaccinology for foot and mouth disease. In this country, we have a stockpile of foot and mouth disease vaccine in case that outbreak pathogen comes back. And of course, you're trying to keep costs down because there's little market. So ideally, you would have a common manufacturing platform that would allow you to quickly switch from one vaccine to another as needed. So we've begun to do a little bit at our own institute, which is the Jenner Institute in Oxford. We have the advantage that we are a partnership with the Preparedness Institute, the largest veterinary research institute in the country, who do a lot on veterinary vaccinology, and that provides critical mass and a lot of know-how and experience and expertise when you remember that essentially all of these outbreak pathogens are coming from animals of one sort or another. This is what we aim to do, not just to make new vaccines. We will never sell any, so we do partner with industry and must, and drive the One Health agenda, the idea of medics and vets working together for common goals. This is our pipeline. We do a lot of malaria, TB, HIV, and so on, as you can see here. Ebola is unusual on this graph of our pipeline because these were not our vaccines. We didn't make them. We helped out in the emergency, as I've described. But we are becoming interested in this problem and are now making some of our own vaccines. Luckily, there is funding. There's a new initiative that I'll come to in a moment, the UK Vaccines R&D Network. One of the target diseases is one that's not well known, unless you're a veterinarian working in East Africa. It's called Rift Valley fever, spread by the same mosquitoes that spread Zika. Aedes mosquitoes affect a wide range of animals and then can infect humans. This is George Wirimwe, who leads the program. He's also potentially susceptible as a Kenyan vet. The distribution, the appearance of this virus is classic outbreak pathogen. These are the outbreaks, different parts of Africa in different years, depending, frankly, on the weather and the rainfall and the number of mosquitoes. And the bad news is that this year is not supposed to be a good year. There's a major Rift Valley fever outbreak predicted, with increases in the number of mosquitoes and probably an outbreak in some of these regions. Can you make a vaccine for that? Well, using the same principles I described for Ebola, the answer is yes. George has done this. Put it into chimpanzee adenovirus, taking the coat protein again. You get neutralizing titers in sheep, in goats, in cattle, and interestingly, in camels as well, who are also infected. And when these animals are challenged, this was done at Ilri in Kenya, the vaccine efficacy is very high indeed. MERS, same sort of problem, but this time coming from camels. 1,600 deaths, 1,600 cases, 580 deaths in 26 countries, largely in Saudi Arabia and spreading from there. Camels are clearly the source of this zoonotic infection, and it's been chosen as a priority disease for an outbreak pathogen vaccine by WHO. Camels are largely infected by this virus. The older animals are seropositive and healthy. The younger animals become infected, shed virus, and infect the camel herders. So maybe the right thing to do is to vaccinate camels rather than healthcare workers, or healthcare workers and camels, more likely. Again, a single-dose immunization, will it produce a neutralizing antibody response, protection, will it be thermostable, etc. And so far, we can show you some nice data on neutralizing antibody titers against the MERS spike protein with a candidate vaccine of the same type that was used against Ebola, a chimpanzee adenovirus vector. This is Sarah Gilbert's work. And then my third and last example is a very topical one of Zika virus disease. So Zika is caused by a flavivirus, a different type of virus, not a filovirus, not a bunyavirus like Rift Valley fever, discovered by accident in Uganda in 1947. Wasn't noticed to cause disease until some years later in Nigeria. And of course, as we all now know, causing microcephaly, very likely, not absolutely certain, but I think it's very likely indeed that it's causing microcephaly in South America and Central America. Occasional cases of Guillain-Barré syndrome, not just in the Americas, but also in the Pacific Islands, where it spread from into South and Central America last year. And of course, this is now being declared a public health emergency of international concern by WHO, just as Ebola was over a year ago. But to be clear, there's a big difference between vaccine development for Ebola and Zika. People have been working on the preclinical development of Ebola vaccines for a decade, supported by biodefense funding. Zika, we're starting from scratch. No candidate vaccines, no animal model, so we can't test this in animals yet. Not clear who you would want to vaccinate. We need better epidemiology. Everybody, women of childbearing age, when they're pregnant, before they're pregnant, the men as well. Can we do this? Well, the good news is there are flavivirus vaccines. You may have had some of them. Yellow fever vaccine, Japanese encephalitis vaccine, manufactured in the UK, as it happens. A recently licensed dengue vaccine in Mexico and now Brazil. And research on hepatitis C and West Nile vaccines. So is that good for Zika? Will it be easy? Well, probably not. That may not be particularly quick. So you have a genome here with a coat protein known as the envelope protein that you might target, and lots of people are. You might try and get antibodies against that, or even T cells against the non-structural proteins. All of those strategies are being explored and might work, but we don't know which will. And in a sense, the race is on. At least 10 groups around the world are trying to make a Zika vaccine. NIH, various companies in India, Hawaii, and so on, the same company that made the VSV vaccine for Ebola. Butantan, you heard about earlier. We're having a go with the chimpanzee adenovirus, the same idea as I described for the other approaches, but we have just started. Maybe Sanofi will be using their platform that they used for dengue and others as well. And so the question we're always asked by journalists is, how long will it take? When will it be ready? And the bad news is that most vaccines typically take a decade or more to develop, but then the Ebola vaccine took 9 months, but it was made already and in a manufacturing lot. For Zika, I've done a back-of-the-envelope calculation. For those of you who like the numbers, they are there. I can't see how you could do this in less than 18 months. You might get into the clinic by the end of this year, assuming that you've started already, but that's into a phase 1 approval and vaccinating the first people. Then you have to do your phase 1, and where you do your phase 2, phase 3, and how is far from obvious. So it's not going to be very quick, I'm afraid. But the good news is that this field is waking up. There is a field at last. People are making vaccines, as you saw for Zika. The UK has more than played its part. The Prime Minister at the G7 summit in Berlin in the summer announced an initial £20 million. That grew in December to £120 million, which is a fantastic injection of funding. And there are other agencies, of course, in other countries very interested in supporting this. It does need global coordination. Not clear how that's going to happen. So let me finish with the financial issues in a little bit more detail. It's all very well being able to make a vaccine, but are people really going to pay to get it licensed? And that is a lot of money. And if you're going to invest in it, is there any other way that you might make a business case? Well, I've mentioned one already. if there's a lot of funding around for biodefense, and there was after 9/11, there was a huge Project BioShield in the United States, you might get a rich government to fund your vaccine stockpile lest there be a bioterrorist attack. And that certainly has helped the field. That funding is diminishing a little bit. You'd like to think the threat of bioterrorist attacks may be diminishing as well, but it's not entirely clear. But there's a second idea that's more novel. It's not my idea. It's been discussed a little bit, and I think it would be really good. So let me run it by you. This would be a global biosecurity outbreak vaccine fund. So to be clear, this is not the vaccine funding we use to vaccinate babies in the developing world. Gavi and those funds exist. They do a very good job, and most babies are vaccinated every day in the world with up to 10 vaccines. That's a very good example of public investment in public health, but I would argue this is public investment in public security. So the idea is very simple. You don't vaccinate everybody with Lassa, with Marburg, with Ebola, with every vaccine on that list. You choose your vaccines according to what's likely to happen in that particular region. So in China, you'd prioritize EV71, influenza. In West Africa, it would be Lassa and probably now Ebola, and so on. And the idea is you would protect by vaccination the frontline workers in the relevant area who are at risk. So this would be nurses and doctors, probably burial teams as well for some pathogens, obviously to protect them. But what's less obvious is that work by various people have shown that these healthcare workers play an important role in transmitting that virus into the community. If they get infected from a patient who doesn't know, nobody knows is infected yet, they go back to their household, transmit, and suddenly the outbreak starts. And you can show by mathematical modeling analysis that if you could stop the first responders getting infected, you'd stop a lot of outbreaks. So you can do that analysis and see if that's a worthwhile investment of time in vaccinating these individuals. And I think the bottom line, in short, is yes, you would protect these individuals and you would protect the community. And if you stopped one outbreak, like the Ebola outbreak I've been talking about, you'd save the order of $5 billion. So suddenly the numbers start to add up. You'd need to keep these individuals vaccinated all the time, so a durable vaccine would be very important, and some of the vaccines I talked about look more durable than others. You'd probably be giving multiple vaccines to high-risk workers, but that's relatively easy to do because these healthcare workers are around and more accessible than the whole population. So this idea, I hope, is gaining some traction, but it's nowhere near happening yet or agreed by any of the funders who would fund it. Finally, vaccine manufacturing capacity. I was asked by the House of Commons Select Committee how long it would take, how quickly could we make enough Ebola vaccine to protect all of us, the UK population, 60 million people. To which the sad and unfortunate answer is you can't, at least not in the UK. We have very little vaccine manufacturing capacity. We do manufacture some licensed used vaccines in facilities owned by an Australian and a French company, not very large facilities, for two particular vaccines. Then we have a bit of contract manufacturing capacity, which isn't very, very high scale. So if you wanted to make 60 million doses, you'd need to build the facility. That doesn't happen easily. It isn't cheap. It would take years to build and years to validate. So if the UK wants to protect itself by having vaccine manufacturing capacity, it's about time we started. And I would certainly recommend that. So this is a particular challenge for some countries. It was a big challenge during the Ebola outbreak. These were new types of vaccine. As I emphasized, the chimpanzee adenovirus hadn't been used for anything before, neither had the VSV vaccine. We knew how to make small lots, but that's very different to being able to make tens of millions of doses cost effectively. That process doesn't exist in most places yet. So the scale was modest, and the people doing the manufacturing did a fantastic effort, actually, but it wasn't easy. The UK is particularly challenged by not having much vaccine manufacturing compared to, say, Belgium. And it takes time to rectify. So if we want to address that, now is the time to start. So in summary, I think there are three key needs. We need to develop vaccines against many outbreak pathogens that could cause outbreaks like the one I've been talking about, and make stockpiles available. And some money is appearing for that. We should probably deploy some of these regionally, as I described, to reduce the risk of outbreaks, not only affecting the healthcare workers, but spreading into the community. And we should build more vaccine manufacturing capacity, especially in the UK. And of course, somebody's got to pay for this. Who might pay? I'm not going to give you the answer because I don't know the answer. I don't think anybody does. But there are a list of suspects, and prospects perhaps, who might contribute to this. So I've put them on this slide to stimulate discussion. International development agencies pay for a lot of vaccines. The World Bank, run by a medic, Dr. Kim, is interested in this problem. Maybe he can come up with a financing solution. Major charities like Wellcome played a fantastic role in the outbreak response, and Gates very interested in this. Or, to go where the money really is, defense budgets. Is this really a health issue, or is it a biodefense issue or a biosecurity issue? And of course in the United States, a lot of vaccine development is done by the military. National governments in high-risk countries are put lower on the list. Most of these can't afford to pay. And large vaccine companies have done a lot in Ebola. May not be so keen to do more if they are going to lose money every time. We may hear about that. So in my last slide, the conclusions are that rapid clinical trial responses to outbreak pathogens are possible. The Ebola response was unprecedented. It was extraordinary. It was life-enhancing for those of us who had the privilege of being part of it, and we tried to help as fast as we could. To be frank, we'd love to do all our vaccine trials as quickly as that. We aren't allowed to do that. There are more administrative burdens. There are a lot of other viruses out there, threats against which we have no licensed vaccines. We need to wake up and do something about this. And in short, we need some new vaccines, new deployment strategies, innovative ways of using these vaccines so they're best used, and more manufacturing capacity. So in conclusion, I would like to show, rather than read out, the names of quite a few of the people, but a small fraction of the people who have contributed towards this effort. In our institute in Oxford, doing some of the preclinical work, the clinical trials team who downed tools from doing malaria vaccines and started doing Ebola pretty well overnight and did a great job. Fantastic partners at GSK, at Janssen, at the NIH, another company, Bavarian Nordic I've mentioned, WHO playing an important coordinating role, other sites in Africa, and critically these 4 funders who very rapidly responded to the need to do something and helped out when we needed them most. Thank you very much for your attention.